Good morning. It's December 17th, and this is your daily brief in science. Here's everything you need to know. A new AI tool called V2P links genetic mutations directly to the diseases they are likely to cause, a development that could speed and sharpen diagnoses for rare conditions. Built by researchers at Icahn School of Medicine at Mount Sinai, V2P moves beyond broad categories to predict which diseases specific variants may drive, with plans to refine toward individual diseases and incorporate data on gene interactions and protein function. Trained on a large database of variants with known outcomes and tested on de-identified patient data, the model often ranks the true disease-causing variant among the top 10 candidates. The work is published in the December 15th online issue of Nature Communications under the article titled Expanding the Utility of Variant Effect Predictions with Phenotype-Specific Models. Contributing researchers include Meltem ECE Kars, Baptiste Milisavljevic, Matthew Mort, Peter D. Stenson, Jean-Laurent Casanova, David N. Cooper, Bertrand Boisson, and Peng Zhang, among others. Senior authors say V2P could streamline diagnosis, shorten diagnostic odysseys for unexplained conditions, and guide targeted research and treatment strategies. The study also highlights quotes from first author David Stein and senior author Dr. Avner Schlesinger about faster, more accurate, and clinically relevant results for rare diseases. Funding comes from NIH grants and foundations such as Fondation Leduc and the Helmsley Charitable Trust, underscoring strong financial backing. The findings emphasize translating genetic changes into disease pathways to inform diagnostics and treatment strategies, advancing the clinical utility of genotype-to-phenotype predictions. Overall, V2P represents a step toward precision medicine and could shape future research and clinical practice in rare disease diagnosis. A new approach to mRNA therapy uses the cell itself as the selectivity valve, redesigning the mRNA payload so it activates only in cancer cells. Researchers showed that this cell-selective system can broaden mRNA therapies beyond cancer and reduce toxicity by relying on intrinsic cellular patterns rather than delivery methods alone. The work, led by scientists at Icahn School of Medicine at Mount Sinai and published online in Molecular Therapy, describes CSMRTS, a 2-mRNA system that acts as a cancer cell-driven on-off switch. One mRNA encodes CAs6 with cancer microRNA recognition sites. The other carries the therapeutic gene with a hairpin. In cancer cells, CAs6 is silenced and the therapeutic is activated, while healthy cells keep CAs6 active and suppress it. In mouse cancer models, systemic delivery via generic lipid nanoparticles achieved strong tumor selectivity, with over 100-fold higher gene activity in breast and colon tumors, and more than 380-fold lower activity in liver and spleen than non-targeted expression. The platform is being developed with patents and is moving toward preclinical development and commercialization, with funding from NanTRNA and NIH grants R01HL142768-01 and R01HL149137-01. The approach shifts emphasis from improving delivery to engineering the payload itself for selective activation, with potential applications across cancer, inflammatory, and metabolic diseases. In studies, tumor growth reductions reached 45% with a tumor suppressor gene P10 and up to 93% when combined with mRNA-based immunotherapy in mice. The work highlights microRNA patterns as internal switches that enable selective gene expression while reducing reliance on traditional delivery vehicles. OpenAI has unveiled Frontier Science, a new benchmark to test PhD-level scientific reasoning in AI across physics, chemistry, and biology, featuring Olympiad-style and research-style questions written by experts. GPT 5.2 leads the pack, scoring 77.1% in the Olympiad tier and 25.3% in research, with notable gains over GPT-5 in the latter domain. The benchmark aims to reveal where AI excels or struggles in advanced scientific reasoning and to guide researchers and enterprises integrating AI into scientific work streams. Industry and academia acknowledge progress, but caution that models still struggle with exploratory, hypothesis-driven workflows and can hallucinate on edge cases, requiring human oversight. Frontier science also touches licensing, subscriptions, and monetization of frontier scientific models, with firms like Deloitte noting potential reductions in R&D costs. Analysts project AI could drive meaningful economic value by year 2030, with industry reports forecasting growth in AI for science and lower R&D costs through AI. Key challenges include data privacy, the need for federated learning, and regulatory considerations such as transparency mandates under the EU AI Act for high-risk AI applications. Ethical and regulatory concerns emphasize diverse training data to avoid biases and the need for transparency in high-risk AI systems. Real-world lab adoption is underway, with pharma and research collaborators using AI for triage and rapid hypothesis validation to accelerate discovery timelines. Potential solutions point to hybrid approaches that combine large language models with specialized scientific tools and symbolic reasoning. The outlook envisions increasing AI independence in hypothesis generation by around 2027, with improving task success but persistent gaps in interdisciplinary reasoning, favoring hybrid human-AI workflows. Skepticism remains about LLMs generating genuine discoveries, with experts warning about unreliable outputs and the need for reliable, expert-informed evaluation. A new light-driven method uses visible light to polymerize water-soluble monomers, creating conductive plastic electrodes without hazardous solvents or UV exposure. Researchers from leading European universities demonstrate aqueous chemistry that avoids toxic reagents, making fabrication safer and more scalable than traditional polymer electrode methods. Water-soluble monomers polymerize under visible light, eliminating the need for strong or toxic reagents and ultraviolet exposure. The work, published in Angewandte Chemie by Tobias Abrahamson and colleagues, highlights broad potential for medical devices, diagnostics, and wearable tech due to its simplicity and surface-level versatility. Electrodes can be formed directly on glass, textiles, and skin, enabling flexible, surface-localized electronics and biocompatible sensors. The approach removes hazardous solvents, paving the way for large-scale manufacturing of organic electronics and sensors integrated into garments or other surfaces. The study points to wearable sensors in garments and broader medical and renewable energy applications enabled by solvent-free processing. In vivo tests on anesthetized mice showed patterned electrodes on skin with improved low-frequency brain activity recordings compared with traditional metal EEG electrodes. The method uses simple LEDs and aqueous chemistry, suggesting potential for wearable sensors and scalable organic circuits on diverse surfaces. The resulting material is an organic mixed conductor that carries both electrons and ions, enabling direct interfacing with biological systems and enhanced tissue compatibility. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.